Hello and welcome. I'm Gwen Taylor, Senior Developmental Editor and Webinar Program Manager with Current Protocols at John Wiley & Sons Publishers, and I'm delighted to introduce today's webinar titled Methods for Validating a Gene Editing Approach to Treating Brain Disease with Zinc Fingers, TALs, and CRISPR-Cas9. This webinar is being co-sponsored by Current Protocols and MilliporeSigma. MilliporeSigma is the North American life sciences business of Merck KGaA in Darmstadt, Germany, and they're dedicated to providing scientists and engineers with best-in-class technologies, lab materials, and services with the intention of making research and production simpler, faster, and more successful. Current Protocols is in its 29th year and is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research protocols available for life scientists worldwide. With 18 titles and over 18,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. Your questions will not be seen by any of the other attendees. The webinar will be recorded and available for viewing in the next few days, and we will send you an email with details on how to access the on-demand webinar, along with a customizable certificate of attendance and a PDF of the slides. So now it is my pleasure to introduce today's speakers. David Segal is a professor of biochemistry and molecular medicine at the University of California at Davis Genome Center, with joint appointments in the Department of Pharmacology and the Mind Institute. He received his PhD from the University of Utah in 1996 and then did a postdoc at the Scripps Research Institute, where he helped develop one of the most widely used methods for engineering programmable DNA-binding proteins. He joined the University of Arizona in 2002 and UC Davis in 2005. Dr. Segal's research focuses on targetable nucleases and transcription factors for gene therapy and genomic research, with a particular focus on neurologic disease. Jun Park is a lead application scientist at MilliporeSigma. He studied biomaterials and immunology at the University of Toronto and did postdoctoral work at Harvard Medical School investigating small molecule regulators of beta integrins. Since joining MilliporeSigma in 2006, his primary work has focused on novel detection technologies, cell-based assays, and refining new approaches and perspectives for Western blotting. So let's go ahead and get started with a very warm welcome to you, Dr. Segal.
Thank you, Dr. Taylor, for that introduction, and I appreciate the opportunity to share today some general aspects about methods for validating gene editing approaches and treating brain disease with gene editing tools, and talk a little bit about the work that we're doing in this area as well. It's hard to imagine that anyone watching this webinar would be unfamiliar with some aspects of gene editing, since gene editing has become so ubiquitous in the life sciences now. At the top of the slide, I'm showing 4 different platforms for engineering gene editing tools, specifically gene editing nucleases. Nature's sequence-specific nucleases are shown in the bottom right-hand side of the meganucleases. Meganucleases occur in nature and are very sequence-specific and very good at making double-strand breaks. However, they've been very difficult to engineer, so while they're used for some applications, they haven't found wide use for many applications. The first types of gene editing nucleases that were easily programmable were the zinc finger nucleases, shown in the upper left-hand side. They could be engineered to recognize a great many sequences, although perhaps not every possible sequence, and they were also a little bit difficult to engineer. However, they did sort of establish the gene editing field in that many things, even many things that are done today, were first done with zinc finger nucleases. I would also like to make the point that if someone has a zinc finger nuclease or product that works well, it's a perfectly good nuclease. The next type of technology to come along were the so-called talons, which is another type of DNA binding protein that's found in nature, and nuclease domains were attached to that. It has some structural features that make them more easy to program than zinc fingers, and they seem to work a lot better on the first try. Columns are also very specific and have excellent cleavage efficiency. It does require that a new protein be designed for every sequence that needs to be recognized. The kind of third generation technology, which I think everyone on this webinar will have heard something about, is the CRISPR-Cas9 technology shown in the lower left-hand side. This is a technology that has been extremely easy to use and accessible. It's an RNA-guided protein nuclease. It's very easy to make lots of the guide RNAs, and it doesn't work every time, but it almost works at very high efficiency every time with fairly good precision. So CrisprCas has caught on and spread like wildfires throughout the life science community, allowing people to do gene editing. And the types of things, no matter which of these platforms you use, the types of things that you can do with these tools is to make targeted double-strand breaks. And the targeted double-strand breaks can be processed in a couple of different ways. On the left side, I'm showing a nuclease-mediated targeted mutagenesis approach, which basically just needs the nuclease. Once the nuclease makes a targeted double-strand break in the cells will frequently repair that break with an error-prone process called non-homologous end joining, resulting at some frequency with mutations at the break point. On the right side, I'm showing the nuclease-mediated homology-directed repair pathway. In that pathway, a donor DNA is required and can insert or exchange specific information at the break point. I'll talk about that a little bit later. So for nuclease-mediated targeted mutagenesis, I just want to say a few words about what it actually does. So the repair process, as I said, is a bit error-prone, and so many times when breaks occur, they're resealed perfectly. I think now we know that there are many times that the breaks are resealed perfectly. However, when mutations start to accumulate, they often disrupt the ability of the nuclease to cut at that site again, and so eventually many mutations will appear. But the mutations are a mixture of small insertions and deletions. So on the right side of the slide, you can see some examples of some deletions and some insertions at the break point. And I think it's important to keep in mind that when people use this, for example, to knock out exons in genes in order to knock out the gene function, that they're really getting a mixture of mutations at that site. So I also would like to highlight how close these types of technologies are moving towards the clinic. And in fact, a nuclease doing this type of targeted mutagenesis is in clinical trial right now. That's the work of a company called Segal Bioscience, and it's an anti-HIV approach. So the HIV virus infects CD4 positive cells in part by binding to the CD4 receptor on those T cells, but also to bind to a co-receptor, which is typically a molecule called CCR5. CCR5 is encoded in the genome, and by applying nucleases, and in the case of Sangamo Bioscience, they have applied zinc finger nucleases, they can disrupt the genes that encode for the CCR5 protein. When that protein's expression is knocked out due to the mutations coming from the nuclease, there's no longer functional CCR5 expressed on the surface, and the HIV virus will essentially just bounce off the surface of those cells. In this way, they hope to be able to give a new immune system to people that are infected with HIV. And in the upper right-hand corner, just mentioning a paper from Tobias et al. That came out a few years ago, where they actually discussed the results of some of their clinical trials. And in fact, they are able to see reduction in viral load in treated patients. And this is now being considered as one of the more advanced methods for actually treating HIV. A couple of points, technical points that I would make on this. In order to knock out the expression of this gene, CCR5, you have to knock out not just make a mutation in one allele, but actually both alleles. So in this case, it would require a biallelic knockout to knock out both copies of the gene. So something to keep in mind when doing experiments, because often when people look at mutations, they count out all the cells in the dish, and then they say, well, there's a 30% mutagenesis rate. But that doesn't mean you'll knock out 30% of the protein. Typically, what a number that's often cited in these studies is that of the cells that receive events, any kind of mutagenic event, a third of those cells will have biallelic events. That means that mutagenic events will occur on both alleles. But that's quite a lot of number of cells that receive biallelic events. But it's also important to remember that not all mutations will lead to a knockout. So if there's an insertion or a deletion that maintains the reading frame, let's say a 3-base insertion or 3-base deletion, that might not lead to a knockout either. So in order to see if your gene therapy or gene editing effort actually knocked out the protein that you want, you really have to do a Western. And that's the way that you could tell that the protein is no longer being expressed. And we're going to hear a lot more about Westerns from Millipore Sigma in the second half of this webinar. Now, this type of gene therapy that I just described to you is an example of ex vivo somatic editing therapy. And this is a case where we can remove cells from a patient, treat them in the culture dish to do the gene editing, then reintroduce the treated cells back into the patient. Now, obviously, this type of ex vivo therapy lends itself to some types of diseases more than others. So, for example, many blood diseases, including HIV infecting the T cells of the blood, but also things like beta thalassemia, sickle cell anemia, severe combined immunodeficiency or SCID. These are some diseases that could be treated using ex vivo therapy. And in fact, what you're going to see is that these will be among the very first examples of diseases that are going to be treated with gene editing approaches. Now, I've talked about using nucleases to make one break and showing how that could knock out expression of a gene if you target an exon. It's also possible to use 2 nucleases to make 2 breaks. And when that happens, you can often result in deletion of the region between the 2 breakpoints. So we call this a large deletion. And the large deletion is actually being applied in the case of treating Duchenne muscular dystrophy. Now, this is not in the clinic yet, but it's very far along in preclinical studies in mice. So in muscular dystrophy, there's often mutations in the dystrophin gene that cause errors in the production of that gene. Specifically, there's often stop codons that allow that truncate the protein from being a complete protein. It turns out that especially in this case of Duchenne muscular dystrophy, the dystrophin protein essentially acts as a bridge. And so what's really needed is the N terminus and the C terminus of the protein. And it's been shown that it's possible to cut out exons in the middle. And as long as you have that N and C terminal still attached, the protein can somewhat do its function, and you end up with a very less debilitating disease. The one way that this has been approached is to use 2 types of nucleases to actually cut out exons that have a stop codon in them, allowing splicing around that exon all the time, and allowing a shortened but still functional protein to be made. So just in January of this year, there were 3 papers that came out in Science that I think were really pushing the forefront of, say, systemic delivery of nucleases into live animals. So these authors were using a viral vector, in some cases it was an AAV, to introduce a nuclease, in this case Cas9 nuclease, injecting them into mice, either into muscle or just systemically, and looking to see the level of large deletions made by having the two nucleases expressed. And quite impressively, they were able to see something between 3 to 18% of alleles treated having a large deletion. And perhaps more impressively, they were able to see gene editing effects when they injected systemically in muscles as spatially diverse as biceps, diaphragm, and triceps. So really, gene editing occurring in lots of places. The rates are not very high yet, but more of this is going to be coming. You're going to see a lot more of this. And I should also say that while this might be some very interesting systemic data, delivery to the liver and brain were published by other groups a few months earlier too. So we're going to see a lot more use of nucleases in live animals, and of course that is pushing towards using nucleases in people as a therapeutic. Now those would be examples of in vivo somatic editing, where you would essentially prepare the nuclease in some kind of a vector particle or some other kind of delivery platform, and then put that function into the person. And as you can imagine, that entails a different kind of set of risks and might demand that the nuclease that's put in not only be very efficient at cutting the DNA, but also have very low off-target effects. Now, off-target effects can be thought of in a couple of ways. Most people, when they think about off-target effects, might think that if my nuclease cuts off target, it might mutate another gene. And that's true, but there's a more insidious way that off-target effects can have deleterious effects on the cell. And that is, if a nuclease is targeted to, let's say, that lower red arrow that's on the diagram, but an off-target effect happens at the upper red arrow on the diagram, you could get that large deletion when you don't intend to. In fact, not only might you get a large deletion, but you also might get an inversion. Two double-strand breaks could also lead to translocations, as well as duplications. And so all of these kinds of events could be happening, and usually in studies where people have looked very carefully, they can find events where using two nucleases can actually lead to large rearrangements in the chromosomes. So in thinking about some of the issues with off-target events that we would like to avoid, certainly mutagenesis in other genes is a concern, but also large chromosomal rearrangements is another concern, and that's also sometimes difficult to find. So we would like to have these nucleases work as efficiently as possible and as specifically as possible. There's been many studies on CRISPR-Cas specificity in particular, because the nuclease is so easy to work with. And there's been a number of different types of methodologies that have evolved, each one giving us the ability a little bit more cleanly to recognize where the nucleases are actually cutting, and then report on all the places in the genome where the nuclease is cutting. I'm not going to review those different methods, but I would point you to this recent review that we authored where we looked at 17 different CRISPR specificity studies. From a review of the literature, one of the surprising findings to us was that although many studies identified CRISPR-Cas nucleases that seem to be very promiscuous and cut at many places, essentially every study also identified a CRISPR-Cas nuclease that had nearly perfect specificity. That is to say, in many cases, using different guide RNAs, they were able to find some CRISPR-Cas nucleases that essentially didn't show any off-target activity in their assay. So our interpretation of the literature's view on this is that it's really dependent on the guide RNA, or perhaps we could say it's really dependent on the target site that you choose. So with some target sites or guide RNAs, CRISPR-Cas has many off-targets, while with different guide RNAs, CRISPR-Cas has fewer off-targets. So the choice of guide RNA is critically important to whether you would have high fidelity in targeting or off-targets. In addition to this information about the choice of guide RNA, and the strong advice there would be to try several different guide RNAs and then use one of the third generation specificity assays to just determine which one gives you the best off-target profile. But in addition to using the right guide RNA, there's been several other strategies that I'm not going to speak about now. But those include things like using double nucleases instead of a single CRISPR-Cas, using evolved versions of the CRISPR-Cas protein with high specificity. There are different types of now Cas9 proteins, and some of these other versions of the Cas9 protein or Cas9s from other species have improved specificity profiles. And even approaches such as using, in cell culture, if you could use the Cas9 protein complex with a guide RNA already, that seems to have better targeting specificity and less off-target effects than introducing the Cas9 and its guide RNA in a plasmid. So some combination of all of these improved strategies as well as the choice of guide RNA should be able to produce even CRISPR-Cas nucleases that cut with exquisitely high specificity, even in a genome the size of human. With in a diploid human genome, there would be 6 billion base pairs. So that's really saying something about the state of the art and where we think we might be able to take these platforms in the future. Now, I didn't talk much about nuclease-mediated homology-directed repair. So I would just say that in making the targeted double-strand break in an appropriate constructed donor, it's possible to make large insertions or even very small modifications. So to put in a large insertion, for example, introducing a transgene into a safe harbor zone in a genome, or perhaps putting a GFP tag in frame with some protein that you want to study in the cell, these would all be examples of cases where homology-directed repair could precisely insert some kind of a large insert in a very precise way into the genome. In some cases, you don't want to put in such a large insertion, and you might just want to change a point mutation. So a method for doing that is actually, the preferred method for doing that is to use a single-strand oligonucleotide because there's less concerns about the DNA integrating. And in many cases, it's been shown to be highly efficient to just use the single-stranded oligo. This would be the preferred method, for example, to investigate the functional value of SNPs that might have been identified in some kind of a genome-wide association study or GWAS study. And the kind of hope is that we might be able to go into cells and just change a G to an A in one base out of the 6 billion base pairs in the human genome. And then you would have cell lines that were either unmodified or modified. They would be isogenic in all positions except for the one position that you modified. And that would give you extremely high analytical power to say that any differences now in gene expression or function of that cell really results from the change of that one base. So this is really being put forward as a paradigm for understanding GWAS SNPs. Of course, another use of this in therapeutics might be to try to correct mutations that occur in genes in the body. But let's just think that out a minute, how that might work. Because oftentimes people hear about a mutation in a gene and you might think, well, why don't we just go in and correct that mutation, and that would be a good therapy. But it's important to remember that when geneticists talk about mutations in, for example, BRCA1, the BRCA1 gene that's involved in breast cancer, there's been over 600 mutations that have been identified to cause deleterious effects in BRCA1. And so just graphically, we could say that the mutation could appear here, or in any of these exons in any particular gene. So if we had to actually go in and mutate or correct point mutations all throughout the gene, we would have to have nucleases that were somehow FDA approved at all these positions. And that seems like a task that would not be easy to address. So oddly enough, one of the thoughts about trying to do therapeutic correction for genetic mutations is not to go in and change the actual mutation, but rather to use a large insertion where you could install a kind of mini gene into some early part of the gene so that you would have a gene function that was still under control of the endogenous promoter in the physiological location, but now you can have a cassette that would essentially bring back that gene's expression without having to fix every mutation that's in the gene. This might probably be one of the ways that homology-directed repair could be used to repair genes, defective genes in people. However, the efficiency of homology-directed repair is usually lower than the mutagenic effects of the non-homologous end joining methods. So we'll probably need another breakthrough in efficiency to get it at least tenfold or more higher before we start to see this in the clinic. That's my personal prediction, I should say. Things are evolving very quickly. Now, with all this talk about could we use nucleases in people, of course, brings up ethical concerns as well of using CRISPR or gene editing technologies for other kinds of methods. And so here in this middle panel, I'm discussing in vitro germline editing approaches where an experimental approach might be to remove zygotes or embryos, do genetic manipulations on those in a dish, and then reintroduce those back into a patient to create a new child with altered traits. So this would be the germline editing approach. And there's been a lot of concerns about the growing ease of being able to do this. And so there's been several countries have passed laws or legislation about what kind of work can be done editing in embryos. And just recently, there were very high-level talks in Washington, D.C., involving our national academies of science, the Chinese equivalent of that, and also the European equivalent of that, coming together in a historic way, actually, to talk about the opportunities and challenges and ethical implications of this kind of work. I think generally there's a lot of concerns about this, and work in this area will probably be restricted until we learn a lot more about how nucleases work, a lot more about how all of this might affect future generations, and really have an open conversation about whether this is even something that ethically people would be supportive of. Because I think that editing the gene line right now is not something that is in the interest of most scientists. And although there is, some research that is going on in this area, but I don't think that anyone really wants to see this enter the clinic anytime soon, if ever. Now I want to switch gears a little bit and talk about some of the other uses of these targetable platforms. There are applications in imaging of sequence-based imaging inside living cells. And I'm not going to talk at all about that, but I am going to talk about using these platforms as artificial transcription factors to manipulate gene expression in the cell. So this would be using these platforms in the role of a transcription factor, in which case all of these zinc fingers, Tollins, and CrisprCas can be used as a kind of targetable DNA binding protein. Even CrisprCas, that's typically a nuclease, we can mutate the catalytic residues of the nuclease, and now it's just a sequence-specific DNA binding protein system. And then we can pull the effector domains off of naturally occurring transcription factors. In this case, there's the red crab domain. And the effector domains can be either activating, transcriptionally activating, or transcriptionally repressing types of effector domains. So we could make artificial transcription factors that can activate or repress genes. And now, unlike nature, we can design them to bind to very specific genes and the promoters of very specific genes, and on some level control the expression of endogenous genes in living cells. So I want to tell you a little bit about the work that we've done in this area, and this was published also this year in Molecular Therapy. We've been looking at neurologic disease, and in particular one disease called Angelman Syndrome, which is a rare neurologic disease resulting in cognitive deficits and learning disorders. Children with this disorder often don't develop language more than a few words. They're often also associated with seizures, sleep disturbances, and some characteristic ataxia. There's some very interesting genetics going on, but suffice it to say for this webinar that this disease results from the loss of UBE3A expression in the brain. So due to a strange epigenetic condition, situation called genomic imprinting, this gene UBE3A is usually only expressed from the maternal chromosome in a typical developing brain. But in the children affected with Angelman syndrome, they either have a mutation or a large deletion that eliminates the expression of UBE3A from the maternal chromosome. There is a copy of UBE3A on the paternal chromosome, but it's epigenetically silenced. And not silenced by promoter methylation, but in fact there's an antisense transcript that keeps it silent. A lot of tricky genomics, but just to say that the therapeutic goal in this case is to reactivate the silenced UBE3A allele on the paternal chromosome. So using artificial transcription factor technology, we designed about 10 of these transcription factors to target a mouse model of Angelman syndrome. We designed some transcription factors to directly target the promoter and try to superactivate the promoter on the paternal allele of UBE3A. And we also tried some transcription factors to repress expression of that antisense. And in fact, what I'm going to tell you about today is our successes in using artificial transcription factors to turn off the antisense, and that then turns on the UBE3A. I also want to point out that in these studies, we were also using zinc finger-based transcription factors, but probably the other platforms would do a similar job. So having shown that we could make some transcription factors that could alter the endogenous gene expression in a culture dish, we needed to think about how this might actually be used in a therapeutic sense. And that brings us to the important delivery issue. In this case, we thought that we could deliver our therapeutic protein as protein by attaching a cell-penetrating peptide, in this case, the TAT peptide of HIV. So a cell-penetrating peptide is any kind of peptide that can endow its cargo with the ability to cross membranes. And in fact, many cell-penetrating peptides could drag in, say, GFP or something like that. And we were hoping that attachment of the cell-penetrating peptide would allow us to inject this protein into mice and that it could cross membranes, including the membranes of the endothelial cells that make up the blood-brain barrier. So in the data that I'm going to show you now, we injected the purified protein into mice, into the peritoneum, so intraperitoneal, and so we would get a systemic distribution of the protein. And when we looked in the brains of the mice, I hope you can see from this picture that we saw a very wide distribution of the protein of our transcription factor throughout the brain. So here we're staining for the HA tag that was part of the protein. And in the outline, you can see a sagittal section of the mouse brain. And compared to no treatment, we can really see our protein all over the brain. And I feel that this is in stark contrast to much of the data using viral vectors such as AAV, where typically the distribution of the virus is very close to the site of injection. So that was a kind of surprising finding for us, but very welcome. But it doesn't really say that the protein is getting in and having an effect on gene expression. So for that, we just looked for the reactivation of UBE3A. And in the panels that I'm showing you here, on the left-hand set of panels, we looked at section brain tissue from the hippocampus. On the right side, we looked at section brain from the cerebellum. If it really got all over, we should see UBE3A reactivation in both areas. On the top row of panels are wild-type mice that express UBE3A, which is shown in green. On the bottom set of panels are the Angelman Syndrome mouse model, which expresses only background levels of UBE3A. When we treat with what's labeled as the TAT-R6 protein, which is just like our therapeutic protein, but has some differences in its DNA binding domain, it doesn't target that site and it doesn't reactivate UBE3A. But our TAT-S1 therapeutic protein, shown with the blue binding region, that does reactivate UBE3A. And then down below, you can see some quantitation. The TAT-S1 protein brings up the expression level significantly, maybe not all the way to wild-type levels. And then we see another Western blot where we can see some restoration of the UBE3A by Western blot. Now, looking at all of this data together, you might ask, well, what would happen if we increase the dosage of the TAT-S1 protein? And we're doing those studies now. But in segueing to the second part of this webinar, I would just say that with increased dose response, you would probably need to use a more quantitative Western blot to see the effects of this therapeutic treatment in a dose response sense, because there, you might be seeing small increases in the amount of protein, and you really want to measure that very carefully. So we'll hear more about that in a few minutes from Millipore Sigma. But for now, I would just like to close and say that I've talked about zinc fingers, TALs, and CRISPR platforms that are useful for altering DNA sequences with mutations, deletions, insertions, and substitutions. I've talked about regulating gene expression with activation and repression domains. And something that we're starting to see happen more now is actually altering other types of epigenetic information, such as DNA methylation and histone acetylation. And I didn't get a chance to talk about the use of these technologies in imaging, but there's a lot of other kinds of application areas. So I think some things to look forward to in the future are better methods to find genome-edited cells, altering epigenetic information, using these to create new animal models for disease. There's going to be more evolution of the tools to make them more specific, more active, and conditionally activatable. And of course, we're going to see them being pushed more and more for in vivo use and use as therapeutics. I just want to close by acknowledging some of the people in my lab that have done this work, particularly the work of graduate student Barbara Bayless, Ben Piles, who does our mouse work, and then Henio Jean and Changhua Ren, who've done a lot of our subsequent work. And with that, I would like to turn the webinar over to our next speaker.
All right. Thank you, Dr. Segal. We now proceed to Dr. Jun Park of Millipore Sigma, who will describe practical considerations for Western blotting with particular emphasis on the quantitative Western.
Thank you so much, Gwen, and good morning, everybody. So let's start by talking about quantitative Western. When I talk to the scientists about Western, often the question that I hear is, but the Western blotting is not quantitative, right? I think the answer is it can be, but there are things that need to be properly implemented if Western is to be used in a quantitative way. With that, I think we'd all agree what Galileo once said, measure what is measurable and make measurable what is not so. Often the reality is that Western blotting is the most accessible way of measuring specific protein expression level in tissues or cell lysates. Let me demonstrate. Before this talk, I did a brief search using Google Scholar, and it was very clear that the quantitative Western is widely used in multiple disciplines. Overall, my search results using a quantitative Western in quotation marks resulted in greater than 13,000 publications using Google Scholar. Before moving into greater detail, let's think about how we often go about doing quantitative Western. I would have a control compared to genetically modified animals, or this would be control cell line versus modified ones, with or without treatment. I would obtain Western blot with a beautiful image like this, from which I can obtain very nice quantitative results. In fact, vast majority of the papers containing quantitative Western result will have a figure very similar to the one that I'm showing you here. While I think these are great results, there are certain caveats that need to be looked at in order to get the most out of your quantitative Western and really have the confidence in the accuracy and precision of your results. I think the first important aspect of any quantitative Western is understanding the linear range of your target protein. When only two protein bands are compared, and often this is what we see in publications, it is very difficult to ascertain whether two protein bands are really different or the same, particularly when results are shown outside of the linear range. Here's an example of 2x serially diluted tissue samples. As you can see, while the serially diluted samples gives an impression of protein signal linearly corresponding to the input amount of lysate, the measurement of actual event intensity can be quite different. Here's an actual result from the same blot. You'll notice how, particularly at high input range, measurements loses its accuracy. And in fact, linear range for this blot would be 2.5 micrograms and below. What this translates to in this particular example is that if your measurements are outside a linear range, while you have 2x differences in protein expression between the samples, the actual intensity measures may only show you a 30 to 40% difference at best. I think another important aspect during quantitative Western is to obtain images with proper exposure time. While this is less of an issue when digital imaging systems are used, this can play an important role when using film as a medium. Shown here are 3 images of the same blot acquired at different exposure time using X-ray film. You can see that overall the optimal exposure time would be 20 seconds. However, if one is not careful, signals on film can become easily saturated, and you can see that at 60-second exposure, upper range of protein bands become nonlinear. In fact, going back to what I said in the previous slide, if only 2 bands are compared side by side, there's a good chance that while the target protein expression may differ by 2x, subsequent quantification will not show such differences. In summary, while the longer exposure will help visualize faint bands, we need to be careful about not exposing the film too long for stronger bands. If possible, always take multiple images and choose your linear range accordingly. Now, I'd like to touch bases on normalization and the issue of linearity when you're measuring 2 different proteins. It is important to keep in mind that there are there is a wide variability in the expression level of proteins in any biological system. Here's an example taken from a science paper where the expression level of different cytoskeletal proteins are shown. What is striking is the range that you see from higher expressor, say actin, to a low expressor, say a Rho protein. There is actually more than 2 log differences between these two proteins. This is important because often the internal controls that we use for normalization are housekeeping proteins that are expressed at much higher level compared to target proteins. Let me illustrate this using an example from our lab. Here we see we have 3 Western blots. ERK was our target protein, and we're comparing GAPDH and HSP70 as potential candidates for normalization. GAPDH, I think many of us are aware, as well as actin, is very commonly used as a loading control and used for normalization during Quantitative Western. Now, these plots show band intensity on the y-axis and as a function of input lysate amount. What you see here is that the GAPDH expression level is much higher than ERK in such a way that its linear range differs substantially compared to that of ERK, whereas the HF70 has a linear range that is quite comparable to ERK. What would happen if you are using GAPDH as normalization control and loading your samples outside of linear range? In one case, you can lose your confidence to say that there are no significant differences. In another case, you lose your confidence to say that there are significant differences because your loading control will look the same regardless of your target expression level. In fact, your loading control is no longer a loading control since it cannot reliably tell the difference between the sample loadings. I think in case like this, identifying proper loading control can give you the confidence in your results, particularly if you're dealing with small yet statistically significant differences, say 30 to 40%, between your control and experimental samples. So please always check and ensure that you're using the right control, loading control, for normalization. Now I'd like to give some practical examples that we did in-house a while back. We're interested in investigating protein expression between normal and Alzheimer's brain samples. So we obtained location and age-matched samples and subsequently performed Quantitative Western. This is an example of one of the neuronal proteins we looked at, GSK3-beta. You will notice that how we have established linear range and also ran 3 independent experiments to verify reproducibility. We also done this with GAPDH and established its linear range. Based on these findings, we have done direct side-by-side comparison between normal and patient samples. On the left side, you can see that the loading control we're using produces a linear range for both normal and patient samples. On the right side, you can see the corresponding expression level of our target protein, in this case GSK3-beta, also falls within a linear range. Now, with confidence, I can normalize and quantify the expression level of different target proteins from 3 independent experiments and apply statistical analysis as shown here to demonstrate significant differences for GSK3-beta and GAP43 expression between normal and patient samples. In summary, always titrate your samples to ensure linear range for your target proteins. If you don't have enough sample, and we ran into this issue with Alzheimer's samples, at least try to run samples in 3 dilutions and do upfront optimization with normal samples. If you're using film, take images at multiple exposure and use an image with desired linear range. Please remember that different proteins have different expression levels, so make sure that your loading control, which often is expressed at much higher level than your target protein, is also a linear range. If you see that your loading control just cannot give you the confidence for your results, try different loading controls that are not affected by your experimental conditions. With that, I would like to start my second half of the talk by covering some key factors that can influence your Western blotting outcome. We often bucket these into 4 groups, and let me start by going over the transfer step first. Most of us are familiar with the role of SDS during transfer, so I will not go over into great detail here, but I think the role of methanol is actually less understood. Methanol actually prevents the gel from swelling, so band resolution can be tightly maintained during transfer. If gel expands during transfer, one can observe diffusive band patterns. Another thing it does is to slow down the movement of proteins and increase their residence time on the membrane. Also, it can enhance proteins' ability to stick to the membrane by removing SDS off the proteins. Taken together, this is a general guideline for transfer step. For small molecular weight proteins or peptide below 20 kilodalton, it is generally beneficial to increase the methanol concentration, say anywhere from 10 to 20%, and decrease the SDS concentration and transfer time accordingly. Also, blotting should be performed using 0.2 micron pore size membranes, which has increased the ability to capture small proteins. Now, for larger proteins, say greater than 100 kilodalton, it is the opposite. It is generally beneficial to decrease the methanol and increase the SDS concentration and transfer time. Also, for proteins larger than 20 kilodalton, it is generally recommended to use 0.45 micron pore size membranes. For retention and blocking, it is important to keep in mind the reciprocal effect of blocker concentration and protein signal. I would like to expand on one of the examples shown here in the middle panel. These blots are probed for P53, and what you can see here is that the ability to detect the protein at low expression level is lost with the addition of non-fat derived milk. But at the same time, the addition of non-fat derived milk helps reduce the background and non-specific bands. Remember that while too little blocking will cause backgrounds to go up, too much blocking will cause a loss of signal at the expense of a reduced background. Blockers in general can be divided into protein-based and chemical-based. While protein-based blockers such as non-fat derived milk are very commonly used, they're not chemically defined and can cause variability, particularly if you're using biotin-related antibodies or trying to detect phosphoproteins. Chemical-based blockers are by nature defined and will offer more consistent results. However, there is no real universal blocker. If you're dealing with difficult antibodies, you need to test and find out what works best for you. The third, and in my opinion, the most significant influencing factor is the antibodies. 2 things are important in regard to antibodies: specificity and the concentration at which the antibodies are used. As an example, shown is an antibody with a very low specificity, and you can see that many different protein bands are lighting up. Another important aspect of antibody is to identify the combined optimal working concentrations for both primary and secondary antibodies as shown here. It is clear that while too much of antibody can cause background, the optimal working concentration needs to be determined empirically by factoring in not only the primary antibody concentration, but secondary antibody concentration as well. Last but not least, it is important to recognize the role of chemiluminescent reagent and its interplay with antibody concentration and sample loading amount. In this example, for a given chemiluminescent reagent, decreasing the antibody concentration will decrease the signal as expected. However, when chemi reagents or bearing strengths are used, working antibody concentration needs to be adjusted in an appropriate fashion. For chemi reagent shown at the bottom, 1 to 1,000 dilution is optimal, whereas for reagents shown at the top, the antibody needs to be used at 1 to 10,000 or even lower dilutions. In summary, sensitivity of your detection reagent will depend on the amount of sample loaded and the affinity of given antibody. For the last few minutes, I would like to show you a device that will help reduce the overall time spent on the immunodetection during Western. It is called SnapID, and it can complete the blocking and antibody incubation step in 30 minutes. It utilizes a small volume of concentrated antibodies to speed up the antibody binding time and uses vacuum to efficiently wash the membranes. You put your membrane into a blot holder and secure the holder into the device. Once assembled this way, you can complete your entire blocking and antibody incubation steps in about 25 minutes with the vacuum-assisted wash steps. Probe blots are then visualized using appropriate detection reagents. Some of the results are shown here, covering a wide range of target proteins of different molecular weights. If you're keen about reducing the time spent during Western, this could be an ideal option for you. With that, I would like to thank Gwen and organizers for inviting me today, and I would like to thank everyone who joined us for this webinar. Thank you, and please let me know if you have any questions.
All right, thank you so much, Dr. Park. So let's go ahead to the question and answer segment. So if you haven't yet submitted a question for Dr. Segal or Dr. Park, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's see what questions have come in so far. This first one may be for you, Dr. Park. First one says, do different blotting membranes affect the quantitative aspect of Western blotting, and do you have any recommendations on this subject?
Yeah, sure. I think that's a great question. So I presume basically it's referring to two main types of different blotting membranes. So one is PBDF. The other one would be nitrocellulose. In general, actually, both membranes are quite acceptable and they will perform well. But I think it is more in regard to the detection system that one may have access to in the lab. Like, for example, if you are interested in using a fluorescent detection system, there are membranes that are actually better optimized for such application.
All right. The next question then, maybe for Dr. Segal here, I'm guessing. It says, do you think purified proteins are the best method to deliver gene editing tools into the brains of live animals?
Thank you for that question. I think purified proteins with regard to CRISPR-Cas, for example, have been shown to decrease off-target effects, and because the protein itself is able to go away quickly as opposed to if the protein was introduced by DNA or RNA. So in those cases, in the case of CRISPR-Cas, purified protein is a very interesting and desired method to deliver the gene editing tools into the brain. However, more generally, I think we're going to have to see a number of different types of methods applied. Sometimes purified protein might be useful, as was shown in our publication, but I think viral vectors and also lipoproteins, nanoparticles, those kinds of things all have a role to play. And I think time will just tell which methods will ultimately be the most useful for most people.
All right, on to the next question. Again, I believe Dr. Segal might be the recipient for this one. The question is, why would you want to edit epigenetic information when you could just edit the DNA sequence?
Well, again, I think it depends on what you'd like to be able to do. A lot of what's done with nucleases today in trying to manipulate gene expression is to knock out gene expression, is to make mutations in genes and disrupt their function. And in some cases, especially in gene therapy, what you'd like to do is activate a gene's function. And so that's not as easy to do with the kind of just mutagenic effects of using nucleases. Now, you could do that if you were going to use a homology-directed repair, but as I mentioned in the webinar, it might take a little bit longer before we really have a good frequency of homology-directed repair and can make those changes in genetic sequence so efficiently. But in the meantime, another advantage of epigenetic editing is that the results might be persistent but not forever, whereas if you change gene sequence, that's a very long-lasting effect. And again, that might be a good thing if you do it perfectly, but you might not want to do a gene change sequence in all cases.
Okay, we have more questions coming in, so even though we're up to an hour right now, I'd like to go a few minutes over, if that's okay with everybody, and get a few more questions in. So next one, perhaps for you, Dr. Park here. What's the difference between absolute quantification and relative quantification?
Sure. I think that's another great question. So I think in general, when we do a Western, we tend to run lysates. So if you're really interested and keen about getting an absolute quantification, in that case, what you will need is a purified protein. So most likely it will be a recombinant protein. If you have those, then you'll be able to ascertain the amount of your target protein in your lysate. But since in most cases we do not have access to such purified recombinant protein, the most of the numbers that we get is actually a relative quantification because we're comparing our samples that are treated, say, versus samples that are not treated.
All right. Another question has just come in. It says, regarding band analysis of control protein to that of treatment samples, is it better to keep the control protein at a constant value, such as 1 or 100%, and then compare others accordingly?
Yes, I think so, yeah. But again, I think you have to make sure because sometimes your internal control can fluctuate depending on your assay system. So you still have to ascertain it. So say you are actually running at 100%, then you probably want to optimize up front that you are running something at 120 or maybe 125%, just to make sure down the road when you repeat your real experiment that everything will be in a linear range.
All right. Next question, perhaps again for you, Dr. Park. Says, I've been using methanol. I've always used methanol in my transfer buffers, but is it all right to use ethanol instead of methanol for blotting?
Yeah, actually, with that question quite often. So from what we have seen, ethanol is actually perfectly fine. We're not able to see any differences by substituting methanol to ethanol. Also, we have heard from some customers that they were actually using ethanol and they were not able to notice any significant differences. So while we cannot guarantee 100% of the time, but I think it's safe to say that you can actually substitute methanol to ethanol.
All right, I think we have time for one more question here. I believe this is for Dr. Segal. It says, do you imagine being able to edit epigenetic information in specific cells in the brain or all over the brain? What's your opinion on that?
Yeah, so in our study, we were able to distribute our therapeutic protein all over the brain. And in the case of Angelman syndrome, we think that's important because we know that that gene's function is knocked out all over the brain in that disease. But for other kinds of applications, you might want to get into very specific cells in the brain, say just the pyramidal cells in the cerebellum or just the dentate gyrus region in the hippocampus. And using the method that I described, we're not able to do that. But I think it might be possible to be able to get into specific cells if, say, we were able to link, if we, say, introduce the nuclease as or the transcription factor as a plasmid, and then put that plasmid under a tissue-specific promoter, or in some other way, we're able to use the genetics of the particular cell to only allow expression of the gene editing tool in specific cells. There might be other kinds of delivery methods that would be more specific as well. Something like focused ultrasounds with microbubbles is something that's actually used clinically and can deliver nucleic acids and proteins locally in a very focal way. And certainly direct injection into the parenchyma of the brain, as I showed in the slide with AAV, is another region to kind of get a very local distribution of these tools. And again, I think it's really going to depend on what you'd like to do, but hopefully from the webinar, I just wanted to give a sense that there's a lot of exciting opportunities in this area right now, and it's really just in its infancy.
All right. So I guess it is now time to wrap up the question and answer session. So I'd like to tell you, though, that today's webinar has been recorded and it will be available for viewing in the next few days. We will send you an email with details on how to access the recorded webinar, along with the instructions on how to personalize and print a certificate of attendance, as well as download a PDF of the slides. So on behalf of today's speakers, Dr. David Segal and Dr. Jun Park, and our sponsor, Millipore Sigma, we sincerely appreciate your attending today's webinar, and we look forward to your attendance at future events from Current Protocols.